Before asparaginase had a name, asparagine was already everywhere in living tissue. It rested inside wheat kernels, potato cells, asparagus shoots, and human blood. Asparagine is one of life's small building blocks, tucked into larger proteins. Most cells can make it themselves, quietly balancing supply with demand. Yet abundance can become a problem when life, heat, or illness shifts. In the kitchen, asparagine meets sugars on hot browning surfaces. Their reaction creates beloved roasted aromas, crusts, and golden edges. But it can also form acrylamide, an unwanted trace contaminant. In the body, another locked problem appears among certain leukemia cells. Some cannot make enough asparagine and must borrow it from blood. Nature had already built a quiet key for both dilemmas. Across microbes, plants, and animals, asparaginase trims asparagine into safer pieces. It is not a hammer, but a careful molecular pair of hands. The enzyme opens asparagine, releasing aspartate and a small breath of ammonia. So the story begins not in industry, but in living chemistry. In 1806, Vauquelin and Robiquet found asparagine in asparagus juice. The plant gave science both a substance and a lasting name. Chemists separated crystals of asparagine, then wondered how organisms transformed them. By the early 20th century, investigators searched animal fluids for hidden catalysts. In 1922, Giuseppe Clementi reported asparagine vanished in guinea pig serum. His observation was modest, almost buried among other questions of metabolism. The clue waited quietly, separated from medicine by decades of uncertainty. But guinea pig serum would soon become a strange biological lantern. In the 1950s, John Kidd studied transplantable lymphomas in laboratory mice. He noticed guinea pig serum could make some tumors shrink dramatically. Serum from other animals did not behave in the same way. The mystery was powerful, reproducible, and not yet understood. For several years, the active substance remained hidden in the serum. Then, in 1961, James Broome identified the factor as L-asparaginase. The enzyme was starving vulnerable lymphoma cells of the asparagine they needed. A simple nutritional weakness had become a doorway into cancer therapy. Researchers then looked beyond guinea pigs for sources that could be shared. Bacteria offered asparaginase in forms suited for careful purification and study. These microbial sources helped move the enzyme from curiosity toward usefulness. By the 1960s, microbial asparaginases entered the clinic for childhood leukemia. The discovery helped reshape treatment of acute lymphoblastic leukemia around biology itself. It also revealed a larger truth about enzymes as medicines. They could act with extraordinary selectivity, following chemistry rather than force. Decades later, another chapter opened far from hospital wards. At the turn of this century, scientists found acrylamide in heated foods. The finding grew from Swedish investigations after a tunnel sealing accident. The discovery was unsettling because those foods were ordinary and beloved. Potatoes, bread, biscuits, and coffee carried the chemistry of browning. Food scientists asked whether asparagine could be lowered before high heat. They wanted prevention at the ingredient level, not correction after damage. The old enzyme offered a practical answer from nature's own toolbox. Asparaginase could work gently in doughs, batters, and cut potato surfaces. Before browning began, it changed asparagine into a less troublesome form. The golden color could remain, while acrylamide formation was reduced. To see its action, imagine a folded protein as a small cove. Asparagine drifts in, held briefly by precise chemical landmarks. Water joins the moment, and one tiny bond is eased open. The products leave, and the enzyme waits, unchanged, for another molecule. No smoke, no pressure, no harsh attack is needed. Only shape, motion, and the patient language of molecules. That gentleness explained why asparaginase fits so many different settings. In bakeries, it can help loaves and crackers brown more responsibly. In potato processing, it helps address a concern born from frying. In roasted foods, it gives producers another route toward cleaner chemistry. In medicine, it remains part of an extraordinary human story. Children once facing grim odds entered a new era of survival. Families learned that an enzyme from nature could become a lifeline. Yet the molecule itself stays humble, doing one reaction again and again. It recognizes asparagine, opens it, releases it, and begins anew. Its history bridges asparagus fields, animal serum, microbiology, kitchens, and clinics. Few enzymes show so clearly how one reaction can touch many worlds. The same quiet chemistry can improve food, guide therapy, and reduce burden. As industries seek cleaner paths, enzymes offer precision without heavy footprints. Asparaginase reminds us that sustainability often begins with a smaller question. What if nature has already made the key we need next? At Enzymes Bio, scientists and enzyme enthusiasts work to make this biology easy to reach. One platform, many enzymes, no tangled procurement for research labs and growing businesses. A green technology with a long future. Learn more at Enzymes Bio.